and we're going to play a little game called the Immuno Scenarios, where you actually get to build an immune system and then get exposed to different pathogens and get sick. And it's a kind of a little competitive game, because the winner gets 5 extra bonus points. So come prepared to play a board game. Also, next Monday will be a quiz, and it will be over the lymphatic system that we talked about on Monday, and then whatever we get to on the immune system today. So the immune system is chapter 21, it's right after the lymphatic system, and you probably realize that they're interrelated because, for example, the thymus is a gland that is part of the lymphatic system and the immune system, because that is where T cells originate, where they're produced. And then we also have the nodes where lots of different white blood cells, the lymph nodes where lots of different white blood cells reside, and where they are able to fight any potential infection. So one function of the immune system is to distinguish. So if we put the function, distinguish self from non-self. And so we'll talk a little bit about how lymphocytes in particular are checked to make sure that they do not attack normal healthy cells. So potentially you could have lymphocytes, white blood cells being produced that would attack your body cells, and that is not a good thing. And in fact, that is a sign of what is referred to as an autoimmune disease. So being able to tell self from non-self is protective, but also being able to recognize or identify cancerous cells, so cells that are reproducing to the benefit of themselves and themselves only, so that they sometimes cause problems in the body. So cancerous cells, which are our own cells that have become renegade cells, and then also cells that are infected with a virus or a parasite. So for example, the malarial parasite actually gets inside the red blood cells and reproduces itself, so it kind of hides inside the cells. Viruses hide inside of cells, and so it's very difficult for the immune system to be able to find them. And so we'll talk about some mechanisms wherein the immune system is able to recognize those cells that have become infected with a virus. Okay, so those are our function. So when we look at the immune system, we can talk about that part of the immune system which is innate. And innate means not learned. Right? So this means that it does not require any previous exposure or experience. And so it's kind of interesting because the invertebrate organisms only have innate. So if you were to, the immune system in, say, for example, a sea star has been extensively studied, and they have only innate defense mechanisms. They don't have adaptive, right? So did you have a question?
Yeah, I was meaning in terms of the not learned, are you meaning it could be like, oh, automatically know that that's
Yes.
the parasite?
Yes.
Okay.
Yeah. So you don't have to have any previous exposure to it. Okay. And this is as opposed to adaptive immunity. So innate immunity versus adaptive immunity. And adaptive means that it is learned. Right. So exposure can enhance the immune response. And this is the whole reason why we can use vaccinations to help prevent disease, is because we have adaptive immunity. So you can give the immune system a little piece of the pathogen or a weakened pathogen, and it will learn to recognize it, so that if it is ever exposed to the real deal that can actually cause the disease, then that individual will not get the disease. And so it's adaptive. So I don't think that they found any adaptive immunity in invertebrates. So it is only found in vertebrates like fish, right? Amphibians, reptiles, birds, mammals, we're all vertebrates, right? So that's the adaptive immunity. Okay. So if we look at some mechanisms of innate immunity. So we'll talk about innate mechanisms. The first one that is pretty obvious is that we have barriers to infection. And I can't imagine what it would have been like to live prior to antibiotics, where, you know, if you got a bad cut or a bad wound, right, there would be no antibiotics that you could take that would help fight off the infection. So you could essentially get a really bad cut and die from it, right, because of the infection. So it must have been, I would imagine that you had to be a little bit more careful, right, around knives, for example, to prevent yourself from getting cut, okay? So the first barrier is like the skin, right? So the skin has a protein associated with it, which is called keratin, right? So that is a barrier. But we also, the skin also has what is called an acid mantle. And so I've heard, somebody told me that they actually had prescription skin cream that is actually called acid mantle. And so sometimes people have skin infections or skin that needs acidified, right? And so I would imagine that this would protect against bacteria, but it also protects against fungi. So we're exposed to fungus all the time, and you can imagine getting a fungal infection on the surface of the skin and how bad that would be, right? So the acid mantle is important. This is actually due to sweat and sebum. So remember that we had sweat glands and we have sebum, which is sebaceous glands. And so the sebum is more of an oily secretion, also helps to lubricate the skin so the skin does not tear, right? And become more liable to infection. Okay. We also have on the skin a protein called lysozyme. And this is an enzyme that breaks down bacteria. And this is also found in saliva, sweat, and tears. So this is an enzyme that breaks down bacteria. We also produce mucus. And mucus functions, and sometimes they spell it with OUS, but mucus functions to catch microbes. Right. So the mucus that's produced by our respiratory tract has a function. It catches microbes and other debris. And remember that we talked about, or maybe we haven't talked about, but in the respiratory system is kind of interesting because even in the trachea, we produce lots of mucus. And there are little cilia, hairs on the trachea that move the mucus up towards the throat, and then we swallow it, right? So the mucus produced by the nose, right, helps to filter the air. And so that mucus would be a barrier to infection. We also have in the digestive system, so this would be, for example, in the respiratory and in the digestive system, like in the stomach. Right? We also have stomach acid, so I'll just put that. So the stomach produces gastric juice. There's little glands that are in the epithelial tissue that produces stomach acid, which is what type of acid, does anybody know? Is your stomach acid? It's a really strong acid, it's hydrochloric acid. So hydrochloric HCl is the stomach acid. Okay, so those are just some examples of barriers to infection. We also have cells that function in the innate defenses. So we'll just put continued here. So we have cells. And one example of the type of cell that functions in innate would be the phagocytes. And the phagocytes include neutrophils and macrophages. And does anybody remember what type of leukocyte are macrophages derived from? Monocytes, right? So monocytes and they mature and they can develop into macrophages. So these are able to ingest and break down bacteria. And there's two mechanisms that they can do this with, probably more than that, but the two main mechanisms is using lysosomal enzymes. So remember that the lysosome, when we talked about in Biology 231, we talked about the organelles in the eukaryotic cell, and one of them was the lysosome. Well, if the bacteria can just take it in, it can then digest it using the lysosomal enzymes. We also have another mechanism, which is called a respiratory burst. So it's kind of interesting because some bacteria, for example, is resistant to the lysosomal enzymes, and there's like 40 different enzymes, right? And so respiratory bursts use oxygen. And so it's kind of like, well, it is hydrogen peroxide. So if we look at hydrogen peroxide, which is H2O2, it breaks down into water plus oxygen, right? And so this is toxic to many bacteria, specifically anaerobic bacteria. So I'll put anaerobic and aerobic. Let me spell that right. Okay, so that oxygen is toxic, okay? And so that's why you put hydrogen peroxide on your wounds, because it bubbles, right? And specifically deep puncture wounds where you could have gotten bacteria that are living in anaerobic conditions, that helps to kill those, okay? So let's look at some video of phagocytes, and specifically neutrophils. So let's see if I can find my video. Okay.
Neutrophils are by far the most abundant type of white blood cell at 60% of our overall count. These cells are usually the first to the scene of bacterial or fungal infections. This neutrophil is chasing foreign bacteria. Eventually, it catches the bacteria and is engulfed into the neutrophil cell membrane. Neutrophils are incapable of renewing their lysosomes, so after digesting just a few of these, they will die. Neutrophils, along with other cells, find their way around by a process called chemotaxis. Following a specific chemical signal, these neutrophils are guided to the site of inflammation within minutes. Leaving the bloodstream, they migrate into the endothelial cells to reach their point of destination. Once they have arrived, they will begin phagocytosing any bacteria present. Neutrophils suffer high casualties, and the result of this is visible in pus seen around the site of a wound.
That's funny music. Okay, so that video showed at the very end there, it was actually showing a white blood cell leaving the capillaries, getting out into the tissues, and then digesting bacteria. And it mentioned that when you have a wound that has pus in it, that is the phagocytes after they die, and sometimes macrophages after they die. That's what makes up the pus, right? And so sometimes you can actually have abscesses, which are walled off areas of pus, and that actually have to be drained. If you've ever had a cat, cats are notorious for getting big abscesses from bites, right? And those generally will not heal up unless they burst on their own, they rupture on their own, and the pus is drained out, or whether you can take them to the vet and have that lanced and the pus drained out, and then they give them antibiotics for that. Okay, so I just want to show you another video that talks about, this is actually produced by a pharmaceutical company that says that they have used inspiration from the neutrophils to develop their own chemical, which would aid in fighting off infection. Oops.
The sequence of oxidative bursts begins when a bacteria is engulfed by a neutrophil, resulting in the formation of a phagolysosome. Once the bacteria is engulfed, azurophil granules containing myeloperoxidase fuse with the phagolysosome. Delivery into the phagolysosome lumen allows myeloperoxidase to catalyze the conversion of peroxide into hypochlorous acid. Hypochlorous acid then reacts with and damages the bacteria. Hypochlorous acid also reacts with taurine, a natural amino acid, to form N-chlorotaurine and N-dichlorotaurine. These two molecules provide long-lived chloronium ions that also lead to the inactivation and death of the bacteria. These three molecules constitute the host cell's primary oxidative weaponry and provide critical defense mechanisms against invading bacteria. The continuous assault by hypochlorous acid, N-chlorotaurine, and N-dichlorotaurine eventually and irreversibly lead to the destruction of the bacteria.
So that shows the respiratory bursts that are catalyzed, we don't watch that one, catalyzed by chemical reactions. Oops, sorry. I don't want to share, I want to close. I don't want to skip the ad. Okay, where is the escape button? There we go.
Confusing because the
Okay. So let's look at the role of neutrophils and other phagocytes and chemicals that are found in inflammation. So this just shows the macrophage. The macrophage has a unique way of capturing bacteria. It lassos it with some of its cytoplasm, and then it draws the substance in, and then you can see that it fuses with the food, the phagosome, and it produces what is called a lysosome that has food inside of it, and then the enzymes break it apart. So that's the stages of phagocytosis. Okay, so let's look at inflammation. And so I have a handout here, which is just one of your diagrams from your book that is not labeled, so you can practice filling out and labeling the different parts of the immune response. Okay, so inflammation. When you have inflammation in your body, what are some of the signs that there is a problem? Swelling. What color does it tend to be? Red. So we have swelling, redness. Is it hot or cold? Hot, right? And then we also have associated with it pain. So one of the things about inflammation is it actually gets a really bad reputation because of chronic inflammation, right? So chronic inflammation is really bad, but acute inflammation, which is in response to infection, is actually a defense mechanism. So it's actually a good thing that sometimes your tissues get inflamed in response to damage, okay? So if we look at the inflammatory chemicals, these are generally produced by damaged tissues and also white blood cells. So for example, basophils and mast cells, which are another type of leukocyte, produce a chemical which is called histamine. And one of the things that histamine does is it causes capillaries to become leaky, so fluid leaves the circulatory system. So capillaries become leaky, and fluid leaves the vessels. Now this fluid actually has a name, and you might have seen it if you have a wound, right? Where it is actually kind of a liquidy, watery fluid that's generally clear. And this is called the exudate. Right? So the exudate is that fluid that leaves the capillaries. And it has important things in it. It has clotting factors. It also has platelets, right? And then it also has other chemicals that can lead to the more production of this leakiness, and then also the production of pain. Okay.
Is that like if someone has cellulitis and they have the fluid that's kind of on the surface of the skin? Is that what that is?
Well, like if you've ever had like a little, like in your mouth also, like if you have a little bubble on the surface of your skin, and or a blister. Blister. Okay, so if you think about a blister, that's the exudate is in the blister. Okay. Okay. So we also have prostaglandins. And these are produced by damaged tissues. And these cause more swelling, and they also cause pain. So when you look at chemicals, drugs that you can take that will reduce pain and swelling, one of these would be aspirin. So aspirin tends to reduce the production of the prostaglandins, as does other pain relievers like ibuprofen. So ibuprofen and aspirin, right, reduce the production of the prostaglandins. They prevent prostaglandins from being produced. And so that's why they say that those would bring down swelling and decrease the pain. We also have kinins that do the same thing as the prostaglandins. So we have different types of kinins, and these do the same thing. Okay. So those are the inflammatory chemicals. So if we look at a diagram of inflammation, the one that I gave you, that first diagram, okay, this you could write in this, oh, you don't have a little box. You could write inflammation above this, right? And so this blue, these little blue dots, or you don't have them in blue, but the little dots that are surrounding the nail here, those would be the inflammatory chemicals. So that's not, doesn't have a line to it, but you might want to write that in. Okay, so those are your inflammatory chemicals. These are the capillaries, right? So capillaries can become very leaky in response to histamine. The arrow that is shown right here is one part of the inflammatory response, which is called leukocytosis. So when you have a invasion of bacteria, in this case, they're labeled as green. Actually, there is a line to that. Sorry. The inflammatory chemicals, this would be the inflammatory chemicals. So this would be the bacteria. I don't think there's a line to the bacteria. Is there? Maybe this is supposed to be. Anyway, never mind. Okay. Okay, so this is leukocytosis. So leukocytosis is where the leukocytes start to be produced at a higher rate in the bone marrow, and then they are released from the bone marrow. So white blood cells are produced and released from the bone marrow into the blood. Okay. Now these white blood cells, this is a neutrophil. The white blood cells need to know where to go. And so what happens when a blood vessel becomes damaged or the tissue around it becomes damaged is that they sprout what are called adhesion molecules, cellular adhesion molecules, but that starts to slow down the white blood cells. So you can see here that the white blood cell is interacting, it's kind of becoming sticky, and it's sticking to this capillary. And so this right here is called margination. So that is where the white blood cell, the neutrophil, kind of slows down and starts to adhere to the capillary. You have a question?
No, I was trying to read it.
Okay. So what happens is the capillary, there's spaces between the endothelial walls, the endothelial cells. And so this process right here where the white blood cells squeeze out is called diapetus. Okay. And then the last part, this would be phagocytosis. So that's where the neutrophils engulf the bacteria and digest them. So this could be also, if you have internal damage, inflammation can take place. So like if you damage your knee, right, the tissues in your knee, you can get inflammation occurring, and that helps to repair, but sometimes that inflammation becomes chronic. And then sometimes scar tissue forms, and then the joint can become non-functional. So it's really important that that inflammation does not become chronic inflammation. Okay. Okay, so fever is another defense mechanism. So sometimes we look at fever as being a very bad thing, but it is actually a defense mechanism because if we elevate the body temperature, then it actually helps to inhibit bacterial growth. It has to do with, I think, the bacteria taking up, has anybody taken microbiology? Is it bacteria taking up iron or something like that? It inhibits their ability to do that, okay? So we have leukocytes that can produce, in response to certain infections, viral, say for example the flu, the leukocytes can produce chemicals that are called pyrogens. So think fire, think pyromaniac, that's fire. And so it's fire generating molecules, pyrogens. And these pyrogens interact with your hypothalamus. And the hypothalamus is what regulates body temperature. So this increases the set point of the body temperature. And so I think we talked about this in terms of homeostasis, but what happens when you first get an onset of a fever? How do you feel? What happens in your body? So you could be like 101 and you feel cold, right? And so you want to like dress up and get warmer, and that's going to help elevate your body temperature. And you also start to shiver, right? So shivering actually increases the body temperature. So this would be the onset, right? This increases temperature, and it is actually using the skeletal muscles to boost your temperature. Okay. And then when the fever breaks, what happens? You don't shiver, you sweat, right? So you go from being like really cold and under the covers shivering, and then all of a sudden you're like kicking the covers off because you've just broken into a full body sweat. And that means that your temperature or your, it has broken. And what happens is that this decreases temperature in response to the set point returning to normal. In response to set point. So that would be a good example of homeostatic balance. Okay. Now, one problem is that babies have a much harder time regulating their body temperature. And specifically when you dress them up, you know, in synthetic PJs and you cover them in blankets, sometimes their body temperature can get above a certain point, and then that can be bad, right? So what is that magical number that you don't want your fever to go above? 106. I think it's lower than 106. 104? Yeah. So actually, you're an EMT, aren't you? Yeah. So you've seen people at 106?
No.
Okay.
But I think that after that, then the proteins, they do that.
They start to denature,
Yep.
right? Yeah. So you want to be careful if your temperature gets above 104. So there has been evidence that suggests if you're sick and you have a temperature of 101, it makes you feel really crappy to have a temperature, but it actually will help to make you recover faster than if you take fever reducing drugs that will take the temperature back down. Okay. Okay, so you want to think of a fever as a defense mechanism that only sometimes goes awry, but it can be very helpful. Okay, so we also have what are called antimicrobial proteins. And the first one that I want to talk about are the interferons. And sometimes these are abbreviated as the IFNs. Okay. So interferons are really interesting because they're chemicals that are produced by cells that are infected with a virus. And they don't help that cell fight off the virus, but it is like a flag signaling all of the other cells that are in close proximity to prepare for invasion, right? So this is produced by cells infected with a virus. Right? It signals other cells to protect themselves by producing antiviral proteins. So interestingly, interferons have been used to treat some illnesses, specifically some actual cancers, and also some hepatitis B and C are sometimes treated with interferons. They tried to use interferons to treat flu, but it wasn't very successful. And then weirdly, I have to do research to figure this out, but it's used to help people that have multiple sclerosis, because it helps send them into a remission so that their autoimmune disease isn't so bad. Okay, so if you look at the second diagram, I'll try to do a better job on this one. Okay, so does anybody know what viruses are? What are the two components of viruses? What are viruses made up of? And these are the viruses right here. So you could put viruses. What are the macromolecules that make up a virus typically? The outer coat is composed of, nobody knows? Protein. Okay, and some viruses have DNA, some have RNA, so I'll just put DNA in this particular instance. Okay. So some of them are when you talk about retroviruses, they have RNA. So this is the DNA. So one of the things about viruses is that they're not composed of cells, but they have to use a host cell in order to reproduce. Okay, so what the virus does is it inserts its DNA into the host cell and uses the machinery of the host cell to produce more viral particles. So this would be this cell right here is already infected, right? And it's already going to probably die from this infection, right? So this is the DNA molecule of the host cell. And what it is going to do is it's going to produce and we're not going to worry about this. This is the nucleus. Actually, we'll worry about it because we're going to talk about, I think we talked about this. This is messenger RNA. This is a precursor to proteins. So what we see right here is that the presence of the virus turns on a gene. Turns on a gene that is then going to code for antiviral proteins, and these are interferons. Right, so this is DNA. We'll put this as the IFN gene. Right. So that's the interferon gene. It gets turned on. It produces messenger RNA, and then it produces interferons. Okay. So this is a signal to a cell that is nearby. This is the receptor for the interferon. Right. This is the interferon. It turns on another gene, which this is a messenger RNA, which codes for an antiviral protein that blocks the virus from entering. So you can see how the use of interferon therapy theoretically could be very powerful, right? So if you could, like if somebody had a flu or if somebody had a hepatitis virus, right? If you could give them the right interferons, then that would signal all the cells, like in their liver, to start producing antiviral proteins that would block the ability of the viruses to enter and to replicate themselves, right? So that's the idea behind interferons and maybe using them from a medicinal point of view and their significance naturally as well, okay? Does that make sense to everybody? Okay. The other type of antimicrobial protein, so this would be the second type of antimicrobial, the first type being interferons. The second type, oh, there's a tiny little spider here. Is it showing up on there? It is not. Interesting. Okay. The second type is the complement proteins. And these are in the blood. And there's like 40 different types that says blood. Okay, so 40 plus different kinds of complement proteins. And complement proteins function by coming together, and they actually form pores. And they insert themselves into the plasma membrane of a pathogen, of a bacteria, or a parasite, and it causes holes to open up so that eventually the pathogen, like a bacteria, will rupture, okay? So they insert themselves into the plasma membrane of a bacteria, and they create pores. So it would be analogous to like shooting it with a, with a bullet, because it creates a hole in the cell, and then what happens, rather than stuff leaving, it actually causes stuff to move in, right? And it's actually going to cause water to move in, and it eventually causes the bacteria to lysis, right? So bacteria will burst, right? So bursting is called lysis. This says create pores. Bacteria will burst. Okay. It also interacts with other parts of the immune system. So here it coats the surfaces and enhances phagocytosis, right? And it also stimulates the release of histamine so that the blood vessels will become leaky. But this is the one particular pathway that I want you to look at, creating the pores. Okay, so everything that we talked about are adaptive defenses, or excuse me, innate defenses. Now we're going to talk about the adaptive defenses, okay? So the adaptive defenses include the lymphocytes, which are the B and T cells. And we talked about how all of the blood cells are produced in the bone marrow, but the B cells mature in the bone marrow and the T cells mature in the thymus. So there's a diagram in your book that shows that, right? So here, the origination of all blood cells is in the red bone marrow. Then they can migrate to the thymus, and then they migrate out to the lymphatic system, for example, and they take up residence, right, in these lymph nodes, where they filter the lymph of any potential pathogen. Okay, so we'll talk about this process in a little bit, okay? But I wanted to talk about what happens that allows the T cells specifically the T cells to mature. So I want to talk about the maturation of T cells. Okay. When we look at the maturation of T cells, we can talk about the types of proteins that are on the surface of our cells normally, and these are called antigens. So all cells have antigens. And we talked about this a little bit when we were talking about the blood types. These are antibody generating molecules, okay? One specific group of antigens is really important, and these are called the MHC proteins. Has anybody ever heard of these? Okay. This is, MHC stands for major histocompatibility proteins. Right? So histo means tissue, compatible. So think about like the proteins that they might check when they're trying to decide whether to do a bone marrow transplant, or when they're trying to decide whether to do an organ transplant. Are these two people going to be compatible? That's the major histocompatibility proteins. And these are unique to each individual, except for identical twins. Right, so identical twins could have the same MHC proteins. Okay, but generally they're unique to each individual. So you can have similar MHC proteins. So people that are genetically related have similar MHC proteins. People that are not genetically related tend to have very different ones. Okay, so you probably have similar ones to your sister or to your parents compared to somebody that lives across the world. Okay. Okay, so these MHC proteins are used, so here, I'll put this, they're used to present, foreign antigens to T cells. Okay. So if I was to draw like a picture of an MHC protein, here's the surface of my cell. here's my MHC protein. It has a little groove in it, right? And that groove can actually fit a foreign antigen. So I could label this, let me make this, oops, make that red, right? So this is my foreign antigen, and it fits into the MHC protein. Oops, I wanted to use black. Right? Okay. So when a T cell is maturing, it has to figure out how to recognize self from non-self. And so we have two different types of selection. We have what is referred to as positive selection. So this is when a T cell, what it is doing inside of the thymus. So this would be at the thymus. Right? We have positive selection. This is where the T cells must regulate or must be able to identify or recognize. T cell must recognize MHC proteins, right? If it doesn't, it dies. Right? So by chance, we could have T cells that would not be able to recognize, right, your MHC proteins. And so that would not be good to have T cells that can't recognize that. So they die. They actually undergo apoptosis, if you remember that from when we were talking about cell division. So positive selection. We also have negative selection. And so negative selection is where the T cells should not recognize self-antigens as being foreign. Right? So if they do, then what can happen is that those T cells could start attacking your tissues, and that could be a very bad thing, and that could lead to autoimmune problems. Okay, so negative selection, if they do recognize, right, they are eliminated. It dies. Right, so they're eliminated from circulation. So this is what is happening in the maturation of T cells inside the thymus. So in your book, they have a picture of this. Okay, so this is showing the MHC protein, and this is a T cell that fails to identify. So recognizing it is like being able to latch onto it like a lock and a key. So this T cell, and we all produce T cells like this one has to die, right? So that's negative, that's actually positive selection, right? Because the ones that can recognize it are the ones that survive. So secondly, if it recognizes a self-antigen and would actually attack it, then that is the one that has to die. So this T cell, because it is locking onto there, that is the one that undergoes apoptosis and must die. This is the one, it doesn't recognize normal self-antigens, and so it survives and continues its maturation. So it's kind of like going to school, right? They're weeding out the T cells that could possibly cause problems, right, from the body. Does that make sense to everybody? Okay. So let's look at how this antigen-antibody thing works. So here, it's a little deceiving because they're showing one antigen having more than one shape to it. But what this demonstrates is that antigens all have a different shape, and the antibody fits like a key, right? So antibodies are the, they are the lock. The antigen is the key. Okay, so this is a specific shape. So you'll notice that the blue structures here are the antibodies, and they are Y-shaped. So when we talk about antibodies, we say that they have a Y shape. And this Y has two different components to it. This, the Y part that comes down right here is not variable. It stays the same in all antibodies. And then I'm just going to draw a little here. Okay. So it is the ends of the Y molecule that have particular receptors. So I'm going to say that this is the antigen receptor, and it is variable. Okay. Now, when you talk about antibodies, I want to dispel the idea that they are the same thing as antibiotics. Okay. So antibodies are not antibiotics. So what antibiotics are particular chemicals that prevent bacteria from reproducing. So these are chemicals that inhibit bacterial reproduction. And a lot of the time, they're taken from other bacteria because bacteria compete with one another to reproduce, right? In the case of penicillin, it's actually taken from a fungus, right? A yeast that grows on bread. Yeast and fungus competes with bacteria for food. And so oftentimes they'll produce chemicals that inhibit the production of bacteria. And then we can use those chemicals as antibiotics, right? So antibodies are Y-shaped, and they're produced specifically by lymphocytes, specifically by B cells, okay? So these are produced by B cells. Okay. Now, if we look at B cells, B lymphocytes have on their surface antibody receptors. So I'm going to draw my B cell, put a B here, and then I'm going to draw my receptor, or my antibody receptors. Okay, so these are antibody receptors. Each B cell has only one type of receptor. And this is really important because B cells are specialists. And what receptor they have on them is due to kind of a genetic shuffling program. So these antibody proteins are coded for by our genes, but the genes shuffle themselves around. And so the production of these antibodies is random, and it is very variable, right? So antibodies are kind of randomly produced. By gene shuffling. So they're very variable. And that is important because it is just by chance that we may have a B cell that is produced that will be able to recognize a pathogen in our body. Okay. So if we look at the diagram, if we turn that diagram, the one that I gave you over, okay, it looks like this, okay? So this type of immunity is called humoral immunity. It's humoral immunity. And this is adaptive. It's also called antibody-mediated immunity. So the body humors are body fluids. So humoral just refers to an old term for body fluid, but it is in the blood. So humoral in this case means blood immunity, and it's antibody-mediated immunity. Okay. So those cells up there, these are different B cells. Right? So notice how they have different, it's kind of hard to see, but they have different receptors, antibody receptors on their surface, but each cell has only one type of antibody receptor, right? So you don't see like a dark blue, light blue, and a green on a single cell, right? They all have the same. So this would be the antigen, right? So this could be a pathogen, this could be a microorganism, it could be a virus. It could also be, interestingly, an allergen, right? It could also be a toxin. So some bacteria produce toxins. Bee venom would have toxins in it, and you can actually deactivate bee venom by producing antibodies, okay? Anything that would generate antibodies. So this is the antibody, this is the antigen binding to the antibody. So this is the antigen binds to specific antibody. Okay. So what happens is that B cell is selected, and it's random, right? So it just happens to be that a B cell can recognize that particular antigen, and it undergoes this, which is called clonal expansion. So what that means is it undergoes mitosis. And so it reproduces itself. So it's producing clones of itself. Now these clones will continue to reproduce, and then they specialize into two different groups of cells. So these cells right here are called plasma cells. And their function is that they are antibody factories. And so the reason why they're drawn with lots of rough endoplasmic reticulum is because that is where proteins that are going to be secreted by the cell are produced, right? So here, these are antibodies that are secreted. So we would look, you would have secreted antibodies. Okay, those are secreted antibodies, okay? Now this cell is really important. This is a memory B cell. And this is long-lived, right? So the memory B cells are the whole reason why we can develop immunological memory and why we can remember pathogens that we have been previously exposed to. So generally, even though it seems like it, you don't catch the same cold twice, right? Rhinoviruses are highly they're always mutating. So it's like you catch a cold once, and then it's like 2 weeks later you catch another cold. Well, it's not the same cold because you already have memory cells to that one. So it has to be a slightly different version of it, okay? So this part, this first part right here is the primary response, because we don't have any memory cells until the very end of the primary response, right? So this whole thing is the primary response from here to here, okay? So the secondary response comes when we are subsequently exposed to the antigen again. So this is a memory B cell. And notice how when it divides really fast and rapidly. And so those would be plasma cells and more memory B cells. Okay, so these would be plasma cells And these are memory B cells. Right? And so you might feel a little sick, but then you're like, oh, I thought I was getting sick, but I didn't get sick. And the reason is that really rapidly, your body produces antibodies that keep you from getting sick. So you've all experienced that where you're like, one day you feel like you're going to get really sick, right? And then your next day you feel great. And you're like, oh, whew, right? So that is the secondary response. So this is the mechanism that we kind of subvert in order to produce vaccinations. So let's talk about vaccinations. Okay. So vaccinations are intentionally presenting antigens to the immune system. Okay, so it could be like weakened forms, right? It could also be like maybe dead. They could also be just pieces of the microbes. Right? So it depends upon which vaccination you're looking at as to whether the dead virus that they're presenting to is just a weakened virus. I think the flu is just weakened. Pieces of the microbes, right? We can also vaccinate against toxins, right? So specifically, when we talk about the tetanus shot, right? So tetanus shot, that's vaccinating against a toxin. Okay, so the first vaccination was really interesting historically, and it was the first vaccination that was developed was for the smallpox. And if you don't know the history of the smallpox, the native peoples in North America and South America did not have any immunity to that particular virus. And so when people started landing on the shores of North and South America, it spread the smallpox virus through the communities. And so we had this huge die-off of native people, right? So they actually think that, you know, the Amazon used to be much, much more populated than it was when we first recorded going there. And the reason why is that the smallpox just decimated huge populations of people, okay? So smallpox causes these lesions all over the body. And I don't know if this is a real picture. It doesn't look like a real picture, but it causes lesions. And so people that survive are sometimes really scarred because of the presence of these lesions. So what they used to vaccinate is what is called cowpox. So the story goes that people that milk cows would get cowpox, but it would be like really weak form of it, and then they would be immune to smallpox, right? So people exposed to cowpox became immune to smallpox. So they were that similar. So people did not have the horrible response to cowpox. But so it'd be like sores on the udder, right? They're milking them, they're exposed to them. And so they might get a few lesions and sores, but it wouldn't be like smallpox. So what they then decided that they were going to do is that they started vaccinating people. So the vaccine was was from scraping cowpox lesions and applying it to the skin of healthy individuals. We'll put a person to be vaccinated. Skin up, we'll just put healthy individuals. And there's a credible story about how they actually got this vaccine to the New World, so to South America. And the story goes is that there was this person who wanted to take the vaccine, but the vaccine could only be in people that had the cowpox, and it took too long to get across the ocean. And so what they did is they took a group of orphans, and they said that they were going to give these orphans a good life, which they wouldn't have had a good life from where they were from. So they took a group of orphans on a ship. And before they got on the ship, they gave 2 of the orphan boys, it was boys, they gave them 2 of them cowpox. So they scraped off the lesion and they put it on the skin, and the boys developed cowpox rash, right? And then as they progressed across the ocean, they just kept scraping and applying it to the other individual, scraping and applying it to the other individual. And so that by the time they got to South America, they had 2 children that were currently had smallpox. And so then they just set up big clinics where people would just come, they would scrape and apply, people would get it, and then they would pass it. And so they essentially just vaccinated themselves, right? And supposedly it saved, and historically, it saved huge numbers of people from this kind of scourge that has decimated the populations previously, okay? So that's the kind of the story of the vaccines and why vaccinations are so important. So if we look at what happens during vaccines, we're actually using that primary and secondary response, okay? So this is my primary response. So let's say that I get a cowpox vaccine, and I start producing the antibodies. And you'll notice that it takes a while for me to produce the antibodies. And so I'm going to get some lesions, right? And then I'm going to start producing the antibodies, and then I'm going to get better, right? So I'm not going to need the antibodies anymore. So then when I'm re-exposed to, say, real smallpox, what is going to happen? What is this called? When you get a second exposure, or maybe sometimes you get a second vaccination. Does anybody know what the second vaccination is called?
Booster.
A booster, right? And so this boosting effect is on the antibodies. So if you look at this, this is the antibody concentration in the blood. So this would be like being exposed initially to cowpox, and then, ooh, I'm exposed to smallpox. Well, my body produces a huge amount of antibodies over a short period of time. So it fights it off, and then I do not, and it keeps it up high, and I do not get actually ill from the real disease. Okay. But notice if it's a different version of it, which happens, say, in the flu vaccine, notice how I still have to go through that primary response, right? And so with the flu is always mutating, and it's kind of a gamble as to what flu is going to be the worst, right? And they actually, I just read somewhere that they actually said, don't get your flu shot too early, because they might decide, they might get information like, oh, we need to change the flu shot from the information of people getting sick about what flu virus is going to be the worst this particular year. And sometimes they don't get it right? And people still get sick, but oftentimes they do get it right, and it keeps people, specifically people like children and old people and pregnant people, from getting really sick, and it could be very bad for them. Okay, so we'll stop there for today, and then we're going to play that immuno scenarios game in lab this week. So that will directly take this information that we've talked about and apply it.